It is my pleasure to welcome you all here to Yogesh Budhathoki's defense. I have had the pleasure of working with Yogesh for the last little over 4 years. He came to my lab somewhat interested in what we were doing, and I remember I sat him down and he talked about the work that he'd done in the field with fish and things in the streams. And I said, well, how are you with computers? And I do that with a lot of students, and I give them the option to either stick around in the wet lab or to try their hand at some computing. And Yogesh took on the challenge of diving into the realm of computational biology. And I will say he never turned back. He has really gone in full force into the work that he's done. He has helped tackle a number of challenges that we've faced. And the biggest one that I know you'll hear a lot about today, we faced a time where we were working on a project that was trying to kind of study the evolution of metastatic tumors over time. And we said, what we really need is a giant atlas of all the different cell types and things within these tumors to even be able to label what's going on, because it was so confusing. And we said, well, let's do that. And so he kind of backed up and took a step back and instead took on the role of being a leader for a large consortium that we formed that gathered together all of the existing data and went through kind of a lengthy process of figuring out what is it that we're actually looking at when we look at this data and look inside these tumors. And I think that it has been very impactful. We just got back from a conference where we had scientists actually come to Columbus this year. We hosted it here in Columbus this year. Scientists from all over the world who are studying osteosarcoma, which is our disease of focus. And there was a lot of people talking about Yogesh's work and the things that he'd been doing. The resources that he's generated have been downloaded by other scientists over 285 times. I checked this morning. It is shaping the way that we study the disease, and will continue to do so for some time, in addition to all of the other things that he's done in the lab. But it's been a pleasure to know him. It's been awesome to see him grow. I think he has faced the challenges that always come with science and with graduate school gracefully. He's always been very, very welcoming of feedback and has used that to make him better. And I think what you see today is the product of all of his hard work. I'm excited to hear about it. Yogesh, time is yours.
Right, I'm just gonna quickly check if everybody can hear online before I start. I hope everybody can hear.
Yes, we can hear.
Thank you.
We hear you too.
All right. Good morning, everyone. Thank you, Ryan, for a very good introduction. My name is Yogesh, and thank you all again for being here. And I'm excited to share my work during my PhD. My thesis is titled Discovery Through the Lens of Multiomics. And I chose this title because a lot of the work that I did during my PhD were heavily relied on some of the modern technologies such as single-cell RNA-seq and spatial transcriptomics. And I'll talk about more later on what those means and how we can leverage those technologies. So I will start by just telling a little bit about myself. I'm originally from Nepal. A lot of you already know that. I came to the US in 2018 to attend college. And I attended Berea College in Kentucky, and I graduated in 2022 with double major in biology and chemistry. After my graduation, I joined a PhD program at Ohio State University. And this is what led me to wonderful Ryan's lab. These past 4 years have been some of the most rewarding years of my life, both personally and professionally. And I'll save all the acknowledgements to the end. But I want to sincerely thank you, all of you, for being here and who has helped me along this journey. So before I start, I want to dedicate all of this work that I did throughout these 4 years to all the children and families that have been affected by developmental disease and cancer. Your resilience, strength, and hope has been very much inspiration behind this work. Growing up in Nepal, I have witnessed firsthand experience on how limited access to health can profoundly affect our life. And those experiences have inspired me, and I hope the work I'll share today will bring us one step closer to understanding this disease and develop better treatment and ultimately give hope to patients and their families. So now is the actual science section of my talk. Throughout my time in Robert's lab, I have worked in a lot of projects, but in interest of time, I'll be talking about my main project today. And this was the primary project that Ryan alluded a little bit earlier. And the title of my talk for today is Integrative Single Cell and Spatial Transcriptomic Analysis of Osteosarcoma Reveals Conserved and Distinct Ecosystem Across Sites and Species. I know it's a long title. Let me break it down to you. So I want to start by telling a little bit about what our lab studies. Our lab studies osteosarcoma, which is most common bone cancer in children, teenagers, and young adults. The biggest danger isn't the tumor itself in the bone, but it is actually when, as you can see here, when the cancer spreads to the lung, and it's called metastasis. Once it reaches to the lung, treatment becomes much more difficult, and lung metastasis is the leading cause of death in this patient. And interestingly, osteosarcoma is 10 times more common in dogs than humans, so making dogs an important model to study osteosarcoma. Now, let me tell you a little bit more about cancer biology, right? I want to make sure you understand like basic background on cancer biology. So we used to think cancer is just growing cells, like uncontrolled growth of the cells, but cancer is much more complex than that. Osteosarcoma or any cancer in general have this complex set of functional states of cells, for example, and within a single tumor, there are tumor cells that are just actively dividing, and some are making a lot of extracellular matrix, and some are secreting cytokines to reprogram cells in the nearby microenvironment. And this complexity changes as the cancer cells go through the process like metastasis, when they move from bone to the lung, for example, or when patients are given different lines of therapies. And on top of this, the researchers use different experimental models to study osteosarcoma, such as mice, dogs, and patient-derived xenografts. And we fully don't understand which model best represents human disease. So the question becomes, how do we systematically understand all this complexity? This is where the single cell and spatial transcriptome comes in, right? So single cell is basically, as it sounds, you take individual cell from the disease or cancer, and then you sequence the gene expression program from those cells, and it will be at a single cell resolution. And then on top of that, we also have a spatial transcriptomics, where you can have an idea of where those cells are located in the space. So over here, what I have is a simple plot that is called UMAP, and I'll be using a lot of these plots today. So each dot here is a cell, and then each group of cell is called cluster, and they are grouped together by similar gene expression. So together, these technologies let us study not only different cell types, but also tell us where they are in the space and how they interact within the tumor microenvironment. So what we did is we assembled one of the largest osteosarcoma single cell data set that represented human, mouse, PDX, and canine or dog samples or patients. And this project involved collaboration of 6 different institutions across the US. And we included both primary and metastatic tumor, primary being bone and metastatic being lung. And then we included, as I said, single cell, single nucleus, and spatial transcriptomics data set. And then we had over 100 samples and over 1 million cells. So in combination, the result of this rigorously annotated resource is called OsteoCard, which stands for Osteosarcoma Exploration and Cell Type Annotation Resource. This tool is freely available to everyone in the community. That's what Ryan was saying. And people are using it to understand, better understand osteosarcoma, explore the hypothesis-driven question. And I will say that it has been a privilege leading this cross-institutional project, and it has helped me grow a lot as a person and a scientist. So ultimately, a comprehensive resource like this, I didn't know that, but yes. We hope that resource like this can help the field move forward. And now that I have introduced you the resource that we created, I will show you step by step what are the things we learned during this process of making or coming up with this resource. And throughout the rest of my talk, I'll be using this workflow here, or figure, if you may, to guide you through the different steps of analysis and what we find during that analysis. I won't spend too much time getting into some of these computational details. Instead, I'll just focus on discoveries that was made during this process.
So we're gonna let this run, see what happens, unless you think you want to keep it going or whatever. I think we're fighting the computer drivers. I think you're gonna help that.
All right, I did it. All right, so basically, when we take tumor samples and sequence them, we not only have tumor cells in the data set, we have a lot of normal cells or nearby cells that was surrounding the tumor, which is all of these called tumor microenvironment. And so the first step when we get this data set is to actually identify what cells are tumor and what cells are non-tumor in our data set. So let me say this again. So you take the tumor and then sequence individual cells. And when you sequence those cells, you do not know what cells are tumor and what cells are not. So the first step in analyzing this data set is to identify in your data set that what cells are tumor and what cells are not. So there are tools, and we began by testing 3 commonly used computational methods, SC-atomic, SCIVAN, and copycat. These are the algorithms that help you identify and most commonly used tumor identification algorithms. And in this heat map, what I'm showing you is result from the analysis. So I'm just using one sample as an example here. And in y-axis, I have the method, and then x-axis, I have each cell index. And red are tumor cells, and orange are normal cells. And if you look at this heat map, you can just tell just with your eyes that there is a high level of discordance in what cell gets identified as tumor versus what cell gets identified as normal. So because of discordance or inconsistency in that we observed across all of our data set, we thought these methods were unreliable, at least in osteosarcoma. So because of this agreement, we developed our own approaches for identifying tumor cells. Matt Cannon developed this algorithm called ScanBit, which is based on single nucleotide variation. And I developed this Thrust ID, which is based on truth set supported tumor identification. And then we tested these methods using synthetic data set where we knew what cells were tumor and what cells were normal. And if you look at this red box up here, that truth is very similar to ATLAS-COL, which is ATLAS-COL is basically this method. So we can say that our method identifies cancer cells at higher accuracy compared to rest of the conventional method where there is high level of discordance on what cells get identified as tumor. And then we used these algorithms on our actual data set. So when we used them, we saw that there was a high level of concordance. That means they were agreeing with each other, further solidifying that our tumor identification method is robust. And we did 2 orthogonal approaches to identifying tumor cells. So now that I have explained you how we went about identifying tumor cells, and we made sure that there was high confidence in identifying the cells, the next obvious step would be to analyze, just take those tumor cells and then analyze them to understand what are the different functional subsets of tumor cells are present in osteosarcoma. And first, when we focus on just tumor cells and group them based on their gene expression patterns, we identified these 6 different tumor subpopulations, which corresponds to color. Each dot here again is a cell, and each group of color is called cluster. Here, basically, are functional subpopulations of osteosarcoma tumor cells. So some of these subpopulations are progenitor-like, such as basal and metabolic progenitor. Basal progenitor are quiescent-like, and they have stem cell-like features, and then metabolic progenitors are, by the name itself, have upregulated oxidative phosphorylation and metabolic pathways. The third one is proliferative. They are just actively dividing tumor cells. The fourth one, fifth one, and sixth one are more differentiated types or subpopulations of tumor cells. Fibrogenic has a lot of extracellular matrix, and fibrotic pathways are upregulated. Interactive secrete a lot of cytokines, and both of these subpopulations are involved in reprogramming. Let me mute all of them. Ryan, can I make you co-host?
Yes, please.
Yes. All right, I just did. Sorry about that. And then the interactive tumor cells, like I said, have a lot of cytokine production, and they're involved in reprogramming the nearby host microenvironment. The last one, the sixth.
Sorry, Yogesh, I accidentally muted you.
But basically what I'm trying to say is there are different sets and subsets of tumor cells that are present in osteosarcoma, and they have specific functions, and they work in synergistic way with each other. So what was even more fascinating was that I just showed you just one data set here in my previous slide, but I'm going to look into both primary bone and metastatic lung samples across human, PDX, canine, dog, and mouse models. We see that all of the colors are matching, at least most of them are. This means that we observed the same fundamental programs that are represented across all these species. There might be change in some of the proportion of these subpopulations of cells from primary bone to metastatic lung sites, but overall architecture of all these tumor subpopulations are very similar. And this finding suggests that osteosarcoma tumors repeatedly organize into a diverse recurring functional groups of cells, regardless of their model species or disease site. So having the tumor cells identified and described led us to investigate what might be happening in non-tumor cells. So while there was a lot of different non-tumor cells here, For today's talk, I'll just focus on myeloid cells, which is comprised of tumor-associated macrophages. Normally, for immune cells like macrophages help us protect from tumors, like by eliminating tumor cells. However, tumor can hijack these normal cells and reprogram our immune system to support tumor growth. And I will tell you more about what we found. So when we look into these tumor-associated macrophages, or TAMs, we found that they were reprogrammed into several functional groups, as shown by different colors here. Some are fibrogenic and some are scar TAMs, and which they are associated with chronic wound healing and fibrosis process. And there are inflammatory and IFN interferon TAMs, which promote, again, inflammation and immune suppression. Interestingly, again, all of these just like tumor-associated macrophages were also consistently observed across both primary and metastatic sites, across both all of the human PDX, dogs, and mouse samples, highlighting their conserved role across different species. One interesting finding was the presence of these green cells here, which are osteoclast-like TAMs, which is found in lung as well. Normally, osteoclasts are typically associated with bone remodeling, and they are not supposed to be in the lung. But somehow, tumor has found a way to hijack the nervous cells and then mimic the bone microenvironment in the lung, just such that there is support for them to grow and thrive. So what this means is that tumor reprogram nearby cells like macrophages. Second, when we treat patient, we should not only think about the tumor cells or targeting the tumor cells, but also the reprogrammed state of non-tumor cells that are supporting tumor. Now, knowing what tumor programs are present or what different cell types are present and what programs they have is a part of the story. The next obvious question would be, in the tumor itself, in the microenvironment, where are they located? And does their location correlate with the function of these cells? So to answer that question, what we did was we took the osteocar, the cell type annotation that I just told you about, and then mapped all those cells into the spatial data set. And it revealed a very interesting pattern. So what I'm showing you here is that 2 samples from human lung patients. Then red here is where the cancer is. And then these are mapping of each of the different tumor subpopulations in the space. So what we found is that at the tumor periphery, we observed this fibrogenic and interactive tumor subpopulations. And as I said, fibrogenic have a lot of extracellular matrix pathways. Upregulated interactives have a lot of the cytokine signaling, and both match with their function. The location matches with their function, meaning that they are actively involved in remodeling the microenvironment in the periphery as they expand outside, right? And in contrast to what we see in the center is this progenitor and proliferative subpopulations, suggesting that the growing states occupy more protected region and serve as a replenishing core. And finally, we identified this subpopulation whose naming was informed by the spatial analysis, and it is called COMA here. It stands for chondroid osteoid matrix associated. And if you look at this histology, there are some gray circles, and these subpopulations seems to be confined within those chondroid osteoid matrix, and they have hypoxic and angiogenic pathway, which is very much stressed microenvironment. So together, these findings show that tumor heterogeneity is not random. Different tumor programs occupy distinct neighborhood within the tumor, and that is very much pertinent to their function. Now, what I have showed you was just focused on mapping tumor cells. So the next obvious step would be to map all of the cell types, including tumor and non-tumor cells. I know this is a very crowded heat map here, but what I'm showing you here is cell type by cell type matrix. The red means that they are interacting with each other or they are close by each other, and blue means they are far away from each other. So this is basically a neighborhood analysis where you can tell what are the distinct neighborhood, what cells are close with each other or what cells are far with each other, meaning that what cells are interacting with each other and what cells are not interacting with each other. So at high level, we see, I'll try to break this heat map down into these actual visualization here. At high level, we see 4 distinct neighborhoods here. The first one being the tumor cells, tumor core itself, which is mostly tumor cells, and some of the other cell type that are being reprogrammed there. And the second one is normal alveoli, which is mostly outside the tumor. And then the third one is the tumor periphery, which is the interface between outside and inside the tumor. And then the fourth one is immune cell pockets. We don't fully understand what these immune cell pockets are doing there and need further investigation. But the major takeaway from these findings is that osteosarcoma tumor is highly organized in an ecosystem with a distinct cellular architecture. So there is distinct neighborhood that osteosarcoma likes to reorganize itself again and again. This heat map is representative of 7 different samples. So now that I have shown you so far, what I have shown you actually is the characteristics of tumor cells and non-tumor cells and where they are located. But then I'm going to switch gears to something totally different now, and that is what experimental models or species best represents the human disease. And this is important question because discoveries that are made in lab using different models is only useful if they reflect the actual human disease. And we can answer that. We can answer this question because we have data set from all these 4 different models. So what we did was we took all these species or data sets from all these species and we compared how do tumor cells communicate with the surrounding across all these different species. So rather than focusing on just the tumor cells, we wanted to include the broader network of non-tumor cells interactions that are happening within the tumor microenvironment. What I'm showing you here is result from our analysis displayed as a violin plot. Overall, this one is dog, this is mouse, and this is PDX. This is primary, and this is metastatic, this is bone, this is lung. So overall, what we see here is dog model seems to be the most representative of human disease across all the pathway modules that is related to cell-cell interaction between tumor and host. So when we look into individual pathways, and again, this heat map shows each individual pathways on y-axis and each species or the model in the x-axis, we see that there are advantages and disadvantages on using each of these species for specific pathways. For example, if you want to study collagens here, all of the models seems to be good, but the dog seems to be the best representative, probably the best model to study collagen pathway. So the major takeaway from analysis like this is to help researchers choose the appropriate model for the pathway they want to study and have the highest translational impact. And for the final, this is the final part of my talk, I want to show you that how people can use this resource of huge cross-species data set. And I know people are actually already using this data set to explore their hypothesis-driven questions and potentially answer them. So in our side, what we did was we asked the question, which tumor host interaction pathway drives metastasis? So surprisingly, when we did analysis on what is the major tumor host interaction that is happening in the lung compared to the bone, we found that fibronectin was number one, although there were lots of other extracellular matrix pathways that is upregulated in lung compared to the bone. This is not normal because lung is mostly air, not extracellular matrix. But tumor somehow reprograms the microenvironment, again, to mimic like bone-like environment again. And again, as I said, one pathway stood out, that was fibronectin, and we wanted to dive deeper into how may fibronectin might be contributing to lung metastasis. Within fibronectin pathway, what I'm showing you here is this heat map between cell-cell interaction. If the cell between, like, for example, this red cell here and this orange cell, which is epithelial cells here, have a big dot, meaning that they are interacting at high level. And all of these red cells here represent different tumor subpopulations, and they seem to be producing a lot of fibronectin, and the major receiver of that signal is epithelial cell, as you can see here in this orange. So I dived deeper, and lung epithelial cells actually seems to be expressing syndican and integrin-like receptor capable of receiving and responding to these fibronectin signals from the tumor. And this suggests that osteosarcoma cells or osteosarcoma tumor cells are actively communicating with the normal lung in the epithelial or normal epithelial cells using fibronectin signals and potentially reprogramming them to support the tumor growth. And the exciting part is that we could see all this happening in the spatial data set. Fibronectin, this is tumor, and then this is fibronectin expression, which is highly around the rim of the tumor. And this is where the epithelial cells are. And when we dive deeper, epithelial cells basically get reprogrammed into this distinct transcriptional phenotype, which are blue cells here. And they have activation of these genes that are downstream of fibronectin. And when we look into what's wrong with this epithelial cell, what are the tumor cells doing to these epithelial cells, we find that they are reprogrammed compared to the normal epithelium, so that they can make a lot more, again, extracellular matrix, they can make a lot more inflammation, and then they are undergoing this transition called epithelial mesenchymal transition. And basically taken together, what I just showed you is that using this resource, we can explore hypothesis-driven question, whereby here what I did was I showed that how might tumor be interacting with epithelium in the lung, and how might they be reprogramming them into more mesenchymal phenotype, so that it can help the tumor grow in the feedback loop. And then there could be some therapeutic opportunities for this pathway here. So in conclusion, what I have shown you today is we created one of the largest and most comprehensive cross-species osteosarcoma resource called OsteoCAR. And using this resource, we identified the conserved tumor programs across different species, and revealed the spatial organization of osteosarcoma within the osteosarcoma ecosystem, and evaluated the cross-species fidelity, or how well do different species models represent human disease, and finally uncovered a potential mechanism involving fibronectin signaling that may contribute to lung metastasis. And all of this work is currently available online as a preprint, and entire OsteoCAR resource is freely accessible for everybody to download and use it. And ultimately, we hope that a resource like this will facilitate collaboration, exploration, and help identify new therapeutic vulnerabilities and accelerate the development of better treatment for patients with osteosarcoma. And now all of this work is possible because I was around a group of people who were very kind and nice to me. I want to take a moment to thank those people. First and foremost, I would like to thank my family. I will say I've been very fortunate to be born to the parents who thought sending me to school was a better idea than sending me to farm, which most of my friends did. To my mother who went abroad for employment to support our education, and my father who worked long hours in farm, and my brother who has stayed home taking care of parents so that I could pursue my dreams abroad. I'm forever grateful. And to my extended families back home, teachers, gurus, and friends in Nepal, your constant support and prayers have carried me through my challenges, and I'm forever grateful for your presence in my life. Next, I would like to express my heartfelt gratitude to Berea College for giving me once in a lifetime opportunities to attend tuition free, for providing an environment that shaped me into a capable, curious, and compassionate individual. To my professor, like Dr. Rosen here, I see him, mentors, host family here, they are both here sitting in the audience, my friends and all fellow Bereans. Thank you for your guidance and for the lifelong lessons that continues to inspire me. And finally, the Ohio State University and the MCDB program. Thank you for providing me with the opportunity to pursue my PhD and for fostering an environment where I could grow as a scientist and a person. I'm grateful for my teachers, mentors, colleagues, friends, and all fellow Buckeyes. Thanks to you that this path led me to the Roberts Lab at Nationwide Children's Hospital, where I found an environment to grow as a scientist and a person. Now, I will say that my PhD was a lot like riding a motorcycle in Maut Ka Kua, which stands for well of death, not in a bad way, in actually a good way. For those who haven't seen what this, or who doesn't know what Maut Ka Kua basically is, it's a show with a giant wooden cylinder where riders race motorcycle sideways along the vertical wall, as you can see here. And it just sounds dangerous, but trust me, it is fun. It is fun. It's loud, thrilling, and every lap pushes you to trust yourself a little bit more. Every lap you make in frustration of failed experiments or analysis makes you stronger. And that's exactly how my PhD felt, challenging enough to keep me humble and exciting enough to keep me coming back to the lab every day. I survived this ride on momentum, caffeine, self-care, some panic, and most importantly, I survived because of the people who made sure I didn't fly off the wall. And first, there you go. All right, he's throwing some money, he is rich. All right, First, Dr. Ryan Roberts, the ringmaster of this Maut Ka Kua. Thank you for welcoming me into the lab and giving me the freedom, trust, and resource to chase my ideas. While I was trying not to crash into the wooden walls of the science, you were standing comfortably at the top of the well, looking calm and throwing down opportunities, support, and confidence, like the enthusiastic spectator who somehow believes that I will survive. Thank you for challenging me to think critically, communicate clearly, and pursue excellence with both independence and curiosity. Working under your mentorship has been highly rewarding experience, shaping my approach to science, leadership, collaboration, and this endless joy of becoming human. All right. Next, Dr. Matthew Cannon, my co-stunt writer throughout my PhD. Whenever coding became terrifying and scary, you didn't stay in audience. You climbed down into the second motorcycle and rode alongside me. While I admire your coding and scientific expertise, what inspired me the most is your ability to meet people where they are, listen carefully, and seek understanding before judgment. Although you judge me nowadays, but that's okay. Thank you for your endless patience and for teaching me how to code. Thank you for always listening and showing me that coding and which apparently is a boring thing for most people. And bioinformatics can actually be fun. And that's what I learned from you. All right. To my lab members, collaborators, funding source, thank you for your friendship, advice, and patience and encouragements throughout this journey. Your willingness to share knowledge, celebrate success, and navigate challenges together has made these years memorable. I am deeply grateful, not only for what we accomplished scientifically, but also for the friendships and memories that will stay with me long after my graduation. Last but not least, thank you to the committee. Thank you for your guidance, support, and thoughtful feedback throughout this process. I deeply appreciate the time and effort and insights you have shared, which have made me and my research stronger, helped shape me both as a scientist and a person. Thank you very much. And thank you very much, everyone, for being here and listening. I'm happy to take any question you might have.
Subhakamana. Subhakamana.
Dhanyawad.
All right, so we have a couple of minutes for some questions. If people want to ask questions from either the online audience or from the room. Your committee will save their questions for later.
Yeah, if there is no question, that's even good for me.
All right, well, let's congratulate.
No, wait.
Is there a hand? Oh, no, we got some hands, okay.
Come on, you're doing fast.
I think this is more of a maybe future something questions because you highlighted using this to be able to analyze where the location of these tumor cells are. But one thing of which I'm wondering, did you look at the different stages of how this cancer develop and how, if the locations coincides with the different stages or they develop as time goes by? Just wondering if you looked into.
Yeah, the question is, do this program change with the temporal relationship, basically what at different stages of the tumor? So based on the data that I have right now, I cannot answer, but I can elucidate into what might be happening based on other experiments, because these data were like fully grown tumor, and we made sure we had enough cells for tumor. But then there are also experiments in our lab that we are doing if at a given moment in time, let's say at the beginning of the tumor, if there is a specific, just one subpopulation of the tumor cells that makes through from bone to the lung. And what we have seen is that that is actually not true. What we see is there is multiple heterogeneous subpopulations of tumor cells that when they migrate from bone to the lung, a lot of them make it to the lung, and then the heterogeneity is diverse rather than just one or two. That's what we have seen.
So great job. I don't think we can understate how much work went into like identifying the tumor cells. Like I remember how much time you spent doing that. One question I had was about sort of the two methods, you know, the one you developed and the one Matt developed, the trust ID and the SNP calling based one. You know, they're a lot more consistent with each other, I think, but I'm sure there are still even differences between the two. How did you handle like discrepancies between those two methods?
Yeah, so we used Matt's method, which is scan-bit. The question was, how did we handle the discrepancy in tumor identification with the two methods that we developed in our lab, if I understand it correct. So ultimately, I decided that my method is correct. So I went with what I identified by using my method as a tumor cells. But most of the tumor cells that were mostly discrepancy between our method were like cancer-associated fibroblasts and then the interactive tumor cells. So, but there are these tumor cells that adapt to the phenotypes of normal cells that might look like normal cells, and vice versa, that there are cells that might look like tumor cells, but they are not. So ultimately, I just had to make a decision and pick that my method is the best and go with it. Sorry.
You've got a question online, Camille? You can unmute yourself.
Yogesh, great job. Where would you go next with this work? If you had unlimited funding, time, and resources, what would be your next steps?
That is such a good question. I have been thinking about moving mostly into clonality studies, basically now adding another layer to this analysis with DNA sequencing, basically whole genome sequencing, and see if each of these tumor subpopulations are actually coming from individual clone, or what is the relationship basically. I have a slide trying to explain that somewhere, but do each of these subpopulation match back to the actual like a clone of DNA, like in a phylogenetic tree, if you look, if they map back to just one, or can multiple clones converge into the same phenotype and vice versa. So that's what I would move next, like adding another assay to this analysis and see if we can identify certain patterns to it. That's what I have thought as of now, but I know there are lots of things that can be done with this data set. Thank you.
Very, very cool.
All right.
All right, thank you very much. Thank you, everyone.
Okay, that concludes the open portion of this exam. So
This concludes.
feel free to grab some pastry on your way out.
Grad student
So
representative here.
basically, this is where all the public will be. The meeting this June. Well, thank you very much for coming. And now the committee will agree. There.
Have one for like a year.
Thank you again.
You have any questions?
To Scott. Thank you. I'll see you guys.
Or certified time get earned.
Yes.
So do you say before?
That's why I asked.
And then we still a lot of emails. Yeah, or conversation.
Got about 50 people online, Yogesh.
Holy goodness, that's a lot of people. 50 people online?
I even have like our collaborators from Heather and Dan, Anand, we're all there.
About this culture here. For new countries.